Hi, everyone, and welcome to episode 110 of Cell and Gene, the podcast. I'm Erin Harris, and my guest for this episode is Yuanhao Qu, a Ph.D. student at Stanford School of Medicine, about his team's innovative work on CRISPR-GPT, which is an AI-powered tool designed to automate genetic experiment design and analysis. He shares how CRISPR-GPT makes advanced genome editing accessible to researchers, even those without deep expertise. And he discusses its origins, the team collaboration behind it, and the development process. We also talk about Biomni, a general purpose biomedical agent that builds on CRISPR-GPT's capabilities to support a much wider range of biomedical research tasks. He highlights the future of biomedical AI, the importance of balancing productivity with biosecurity and ethics, and he emphasizes the rigorous evaluation needed for any new tool. This episode is really cool because it explores this cutting edge AI system that sounds like science fiction, but is very much real. So definitely stay tuned for my conversation with Stanford School of Medicine's Yuanhao Qu. Enjoy. Yuanhao, welcome to Cell and Gene, the podcast. Thanks for being here.
Yeah, really excited today, to talk about my recent works and on CRISPR-GPT today, and really excited to be invited here.
Absolutely. Your work came across, believe it or not, my LinkedIn profile. And as it was, as they say, breaking LinkedIn, breaking the internet. So I was really intrigued by it and thought, I really have to get you on Cell and Gene, the podcast, to put in front of our listeners and our readers of Cell and Gene exactly what CRISPR-GPT is. And so I'm going to let you take it from there. What is CRISPR-GPT and what motivated its development?
Yeah, thank you very much for the question. Really love to talk more about our recent work on the CRISPR-GPT. So CRISPR-GPT is actually an multi-agent large language model system that really automates gene editing experimental design and data analysis process. So just some background, our lab, by Dr. Le Cong, at Stanford, has been really developing a lot of genome engineering technology. Le is also one of the pioneers in developing CRISPR technology when he was back in Broad Institute in the lab of Professor Feng Zhang. So we have been really investigating how to build the best CRISPR system for the past decades. But through the time, we actually have been contact with all different people around the life science industry, to see how they can best perform their gene editing experiments. We have tons of those different collaborations from all different type of areas. That's also where we realize that although this technology has been really amazing and really changing the life science industry, a lot of people may not have the best knowledge on what are the best practice in gene editing and how to perform those sometimes very sophisticated gene editing experiment. So that's really kind of the way that motivate us to build up this gene editing agent called CRISPR-GPT. So we started very early, around 2 years ago, when we happened also to collaborate with one of the AI and CS professor at Princeton University, Dr. Mengdi Wang. So she got the very early idea about we have seen AI and large language model has been really powerful. How about we just combine that with our gene editing technology and try to move things much faster and automate the process about gene editing experiment design and data analysis. So everything started from there, and we started to build up this agent with our original aim about automate our workflow and also enable people who are not really the experts in gene editing to be able to perform the gene editing experimental design and data analysis as an expert way. So after around 1 year or 2 years, especially witnessing the developing of the general large language model, we finally build up this and see how much it can actually empowers individual scientists to perform gene editing experiment even without any experience. So I'm also so glad to see all of this.
Same here, same here. It's so interesting and intriguing and really innovative. And I want to talk a little bit more about the challenges that this CRISPR-based experimental design and analysis you're aiming to address. I know you're talking about, helping giving anyone with perhaps a little bit less experience with, the tool to actually do gene editing in the way that it's meant to be done. But beyond that, what other challenges, like I said, in CRISPR based experimental design, are you hoping to address with CRISPR GPT?
Yeah, as we kind of know, like for gene editing experimental design, there are actually a lot of like key decision points that are critical for the editing experiment successful. On this, we like specifically focus on several key aspects, like from selecting the best CRISPR-Cas system, and then by like selecting the best delivery model or like delivery system, like lentivirus or other methods. We also focus a lot on the guideline design, which is really critical, like in designing the specific guides for gene editing experiments. Besides that, we also have some functionality focused on like the off-target prediction, the protocol generation, and also data analysis in the end. So although I mean like a lot of people right now can actually perform a reasonable like level of standard workflow for all those steps, but if we really want to aim for really high efficiency, we definitely need a lot of like very specific knowledge and data and even tools to achieve those. So that's really why we build up this like gene editing like agent. So as you mentioned, like first, it really enable like people without any experience to perform like a reasonable like gene editing experiments design. At the same time, the power of AI really enable us to achieve like the best in the class performance by retrieving or like utilizing the most up-to-date information on how all of those individual steps are done like nowadays. Uh, so that's basically I would say like what the most critical challenge is addressing right now.
Great, great. Before we move on, I do want to ask you, so you and your team at Stanford, so what is you specifically, what is your role in the development of CRISPR GPT?
Yeah, that's also a great question. So this work is definitely not just like me as a single person, more of like a teamwork. So as I
Sure.
mentioned, like it has like significant collaboration between like our lab, which is really specialized in gene editing experimental design, and also with like Dr. Mengdi Wang's lab, especially Kaishen Huang, who is also like AI CS PhD at Princeton University. So my role initially really helping to design the overall architecture about the agent, like how like we design it into like different steps, and how what type of tasks are we trying to like achieve, and what tools, what data, and what knowledge we need to build up this very important tool. And later, we definitely also spend a lot of time in evaluating the process and the system because I mean, I would say evaluation is one of the key part for the any type of like AI agent or like AI system development in biomedicine right now. Without like a great evaluation matrix or like evaluation method, it's really hard to measure whether an agent system is really useful or not.
Sure, sure. Um, I want to talk a little bit about how you envision CRISPR GPT evolving in the broader context of AI enabled life science research. Where do you really see this going, especially in our, all things AI world?
Yeah, I mean, that's also a really exciting question for me also. So for my past 2 years, I have been especially focusing on developing better AI agent system to really automate our biomedical experimental design and data analysis process. So we really see like CRISPR-GPT as like I would say like an early pioneer of like building this very specialized AI agent system for biomedicine. But as we know, like there's definitely a lot more area, not just like in CRISPR, but a lot of like other area like cancer, like stem cell, and also neuroscience. We have very distinct like those area and knowledge within biomedicine. So the next question is really how we are going to scale the knowledge that we build from like CRISPR-GPT to other area, like in the end of the day, to really automate every single like individual process about like the experimental design, data analysis, and even hypothesis generation, like to form like a end-to-end like AI scientist system that can really help us automate the discovery in the biomedicine.
Okay, so we also have Biomni. And I would love for you to talk us through Biomni and then what inspired its creation as a general purpose biomedical AI agent, which, we were just talking about, biomedicine. What is it and what's the why behind it?
Yeah, so also really excited to talk about that. So Biomni is first it's a like project at Stanford University. Has been like a lot of collaboration over there, especially with Kexin Huang from Yuri Leskovac lab. Kexin and I, we have been knowing each other for a while, have been talking a lot about like this AI plus biology. So he's actually from more of a computer science background and but also has been working on like biomedicine for a while. So after like a few like development of like the CRISPR-GPT agent, we came actually together and really talk about this. How are we going to build this, as I mentioned, like to scale this like across different area. So that's the reason why we build up this general purpose biomedical agent with the aim of it does not like really focus on like a very specific single area, but to be able to like cover most of different aspect of like biomedicine. So this come to say like really Biomni is really like a project focused on building up a general purpose biomedical agent. It first create like an environment, covers a lot of different areas of biomedicine, including gene editing, but also a lot of others like bioinformatics, biochemistry, cell biology, and a lot more. Like, so it really creates this type of like a Lego box that from this general purpose agent, we can cover a lot different tasks in the biomedical working space. At the same time, we can also utilize it to build up like very specialized agent within the general purpose biomedical agent, like CRISPR-GPT, to really focus on a single area. So I really see these two in the end of the day will merge to like a singular thing that we call like an AI scientist system that covers not only the broadness about like what people are working on, but also a really expert level performance on very specific area.
Good. Okay. Now, given that your work spans both CRISPR GPT and Biomni, those AI agents, how do they actually complement each other?
Yeah, that's also a great question. As I kind of touched like in the last question, so we really see these two are both very important. Think about like first, like for CRISPR-GPT, we're really covering a very specific and focused area of biology. We make it really to the expert level. But for Biomni, we really cover the broadness, as we kind of mentioned, like for the biology that like because I mean like when we actually doing research is kind of hard to really just like you only know one area, but usually it will cover at least like several different topics. So that's where like Biome show its power, So it can not only really help us design a gene editing experiment, it can also help us understand like basically like how that context really happens in the like cancer biology or in neuroscience or in like other type of like biomedical area. And also it has knowledge about like how to like really download the existing data from the online sources, how to understand like a lot of like different literatures across different areas, and how to like broadly write code to analyze the different type of data. So I really see these two, they really like couple well, complement with each other. And in the end, I would really imagine, as I mentioned, like a system that both covers like you can do a lot of different stuff, and also it really works well in a lot of area.
Good. Okay. Now, before we wrap up, what advice do you have for researchers thinking of building biomedical AI agents and or what advice do you have for any cell and gene therapy developer out there listening to this who is interested in incorporating these AI agents into their work?
Yeah. So I really have seen like, or at least like I feel like I witnessed really this the power of these AI agents has been really automating and like democratizing the expertise like within different biology area. It significantly like increased the productivity, at least for me, like a PhD student. But I will also say maybe a lot of like other people, like as research scientists across the biotech and life science industry. So from there, I really see the most important thing is really that like we build up this AI agent, not only just like as a research demo or like a research project, but also enable it to really increase the productivity. So that to be said, it's really important to evaluate how well it actually performs in the field work, in the actual daily job that biologists actually do every day. So like whatever, as I mentioned, like in the very beginning, like whatever like AI system you build, in the end of the day, you need to evaluate well. You need to make sure it's very accurate. You
Mhm.
need to make sure it actually is functional well, like can retrieve very great information. And in the end of the day, I also want to emphasize that because we have been able to build up this like really powerful agent system for biomedicine, there's also potential dual use for these tools. So we might also want to care about like the biosecurity potentially behind this. We don't want it like to harm people. We need to like have also some like guardrails and safeguards for the ethical use and want to protect all the potential like genomics data, genetics data from the human. So we also don't want to like put all of those data into the actual large language model training. So I would say still there's quite a bit of work into really bring all of these powerful agents into like the daily life of like biologists. But I would say like that future, although there's tons of work, I can see that happens very soon. Maybe just within a few months or like a year or two, I would say that will really like revolutionize what we're doing right now.
Incredible. Right around the corner. Well, Yuanhao, thank you so much for your time and your willingness to jump on this recording with me very quickly. The turnaround time was really quick, and I want to let our listeners know that uh, he was very, very thoughtful and knowing that we wanted to get this information out to you, our listeners. So thank you, Yuanhao. This was extremely insightful, and we will be following along to see what comes next with CRISPR GPT and you and your team's work on it. And thank you again for your time. This was great.
Thank you. Yeah, really excited to be talking about all of this today. And yeah, we will continue building up like these AI agents and trying to like like build together and then accelerate the discovery process.
Excellent. Well, and thank you, listeners, for tuning in to this episode of Cell and Gene, the podcast. And we will talk to you soon.